Embryonic stem cells originate from embryos obtained by in vitro fertilization of egg cells. In labs, embryonic stem cells are grown on a layer of feeder cells with all of the factors that maintain them in a pluripotent, undifferentiated state, preventing spontaneous differentiation. One of the most important uses of embryonic stem cells is the study of the mechanisms underlying genetic disorders. More than 100 different lines of embryonic stem cells have been developed so far. Each cell line carries a mutation responsible for a specific disease. One example is the cystic fibrosis, a disease affecting the respiratory and the digestive systems that is caused by mutations in the gene CFTR. To study the molecular mechanism involved in the disease, 12 lines of embryonic stem cells carrying different mutations in CFTR gene have been derived. Embryonic stem cells are used in several clinical trials. In this case, embryonic stem cells are driven to differentiate in specific cell types such as oligodendrocyte progenitors, retinal epithelial cells, or pancreas beta cells. Those differentiated cells are then injected in patients affected by spinal cord injuries, macular degeneration of the retina, and diabetes. The use of embryonic stem cells raises important ethical issues, since their isolation requires destruction of the embryo, although these embryos cannot be implanted. Additionally, in vitro fertilization procedure involves donation of egg cells, which is often painful for women. For these reasons, the use of embryonic stem cells for research and therapy is regulated by internal laws, different among countries. The ethical questions on one hand, and the numerous medical applications on the other, are important issues in the ongoing discussion at the international level about the regulations of the use of embryonic stem cells.